In our last tutorial, we are looking at the digestive system, and we looked at some of the organs. And in this tutorial, I want us to see some other examples of organs of the digestive system. One, we have the stomach, and when you look at its walls, it's made up of muscles. These muscles contract and do two things. As the muscles contract, they churn the food. They contract, and then the stomach walls press the food, compress the food, and then the food breaks down into smaller pieces, what we call mechanical digestion. Also, when the muscles contract, it moves the food through the stomach by the process of peristalsis. In the stomach walls, we have glands that produce secretions called gastric juice. This gastric juice has a number of composition, which includes water. This water turns the food into solution for easy action of enzymes, because enzymes cannot digest, they cannot break down solid food. So water helps turn the food in solution so that enzymes can act on the food. We have also enzymes in gastric juice, such as pepsin, which breaks down the proteins to peptides. There is HCl, hydrochloric acid. This provides acidic medium for the action of enzymes in the stomach. Also, HCl kills the microbes or microorganisms that enter the stomach along with food. There is mucus. This mucus coats or covers the walls of the stomach to stop the protein digesting enzymes, pepsin, from digesting the walls of the stomach. However, if this mucus was not there, these protein digesting enzymes would digest the walls of the stomach and cause what we call gastric ulcers. As you can see, here is the structure of the stomach. At this outlet, you see this outlet, there is a ring of muscles called pyloric sphincter. The work of the pyloric sphincter is to let out the liquid food little by little, to let food come out of the stomach little by little into the small intestine here. So, because already with that, by the time food is in the stomach, it's already mixed with water, so it's already in solution form, so it has to move out little by little so that enzymes can continue to break down the food. Then at this other, this is the inlet. Here we also have another sphincter, another ring of muscles called the cardiac sphincter. This one prevents food from going back into the esophagus. However, you can vomit if there is irritation of the gastric. If you have eaten food that has been contaminated, this sphincter can open and allow you to vomit. But if the food is not contaminated, it will not allow the food to go back. So food will only go out at this outlet here, where we have the pyloric sphincter. Then we come to another organ, that is the small intestine. This one is subdivided into three sections. That is the duodenum, the first section, jejunum, and the ileum. Of the three, the ileum is the longest, and chemical digestion ends at the duodenum. So at these two sections, jejunum and ileum, basically it is absorption that takes place here. In the duodenum, the first section, we have gastric juice and bile. They are released The gastric juice is produced from the pancreas, and the bile is produced from the liver. So they all join the small intestine at the duodenum. But before bile is released into the duodenum, it is temporarily stored in the gallbladder. This bile has two main functions. One, it turns the large lipid globules in food into emulsions of tiny droplets. In other words, it breaks down it converts the large lipid particles into smaller tiny particles so that enzymes can easily act on them. As the particles become smaller, the surface area becomes bigger for the enzymes to act. Also, bile neutralizes the food from the stomach for easy action of enzymes. You know that by the time food leaves the stomach, it has already been mixed with hydrochloric acid, so it is acidic. Yet the enzymes that work in the duodenum, they cannot work in acidic media. So bile helps to neutralize the food and turn the food into alkaline, so that the enzymes in the duodenum can act on the food. Then we come to the role of the pancreatic juice. This has many components. It contains the lipase enzyme that breaks down lipids to fatty acids and glycerol. These fatty acids and glycerol are smaller and soluble, hence they can be absorbed into blood. It contains also trypsin enzyme, which breaks down proteins to peptides. It also contains amylase enzyme. This converts starch, which was not converted by amylase in the mouth, so it converts it to maltose. Then when you look at these two foods, the peptides and maltose, they are still large, so they are not soluble and they cannot be absorbed. So they are further acted on by two enzymes which are secreted from the walls of the duodenum. These are maltase, which converts this maltose to glucose, and then the glucose is soluble and can be absorbed into the blood. Peptidases, these convert these peptides to amino acids. The amino acids are soluble and can then be absorbed into blood. Here we have the summary of digestion. We have the class of enzymes. All enzymes that act on carbohydrates, we call them carbohydrases. And the only carbohydrate we eat as humans is only starch. We eat others like cellulose, but we don't produce enzymes that break down cellulose. So the chemical digestion of starch starts in the mouth by the action of amylase enzyme, which is produced by the salivary gland. So amylase breaks down starch to maltose. Then this maltose moves to the stomach, and in the stomach no action happens on it until it reaches the small intestine, where maltase enzyme will break the maltose to glucose, and then the glucose can be absorbed. Also in the small intestine, if there is any starch which was not broken down by amylase in the mouth, will be broken down to maltose again. There is some amylase enzyme which is produced in the small intestine, which breaks starch that was not broken down in the mouth to maltose. Another type of food, that's the protein, and all enzymes that break down proteins are called proteases. An example of proteases, we have these three. We have pepsin, which is produced in the stomach. This pepsin breaks down proteins to peptides. There is trypsin, which is produced in the small intestine, which breaks down proteins to peptides. Proteins which were not broken down by pepsin in the stomach will be broken down by trypsin in the small intestine. Then we have another peptidase in the small intestine. This converts peptides to amino acids, and then the amino acids can be absorbed. So when you look, chemical digestion of proteins happens in two areas, in the stomach and in the small intestine. And lastly is the lipids, which are acted on by lipase enzyme. This lipase converts lipids to fatty acids and glycerol, which are then absorbed. In short, chemical digestion of lipids happens only in the small intestine, and chemical digestion of proteins happens only in two areas, the stomach and the small intestine. Then the chemical digestion of starch starts in the mouth, takes place in two areas, mouth and small intestine. Then after food being broken down to smaller soluble particles that can be absorbed, then absorption takes place. Absorption simply means process by which digested food enters the bloodstream, and this happens by two processes: diffusion, where molecules move from region where their concentration is high to region where their concentration is low. They move from the small intestine where concentration is high into blood capillaries where concentration is low. The second mechanism is active transport, which is the movement of molecules from region of low concentration to region of high concentration with the use of energy. This is the opposite of diffusion. Here, there are some food nutrients that their concentration is high in blood, but we still need them. So such nutrients are absorbed by active transport. We come to structural adaptations of small intestine to its function. Like all other body structures or organs, even the small intestine have structural features that allow them to carry out their work of absorbing food. One, the intestine, it is long and folded, which provides a large surface area for absorption of food. As you know, absorption is mainly by diffusion, and because the intestine is long and diffusion is a slow process, so by the time food moves through the whole small intestine, all the nutrients will be absorbed. Two, it is surrounded by a network of capillaries which maintains a steep concentration gradient. Concentration gradient, I mean difference in concentration between the intestine and the blood capillaries. So whatever amount goes into the capillaries, it is immediately carried away, which helps to keep the concentration of food nutrients in the capillaries lower than the intestine, so that materials can continue to diffuse from the intestine into the capillaries. The third one, the walls of the intestine have tiny projections called villi, which further increase the surface area for absorption. And each villi has tiny projections called microvilli. These further increase the surface area for absorption. Here is the section of the small intestine, and these are the villi we are talking about. As you can see, the whole walls of the intestine is surrounded by villi, and their work is just to increase surface area absorption. Then we have this extension. This is the wall of the intestine. As you can see, this side is the intestine, then the inside is the walls. So food has to cross this boundary, and you can see the moment it crosses, there is this network of capillaries, this red line you see here, so that by the time food diffuses, it enters in the capillaries, then the capillaries carry the food away, which keeps the concentration of food in this region lower than this region, so that the materials can continue to move by diffusion. So this is an extension of one villi. As you can see, these are the microvilli, which further increase the surface area. Then we come to the last section of the alimentary canal. That's the large intestine. As we have already seen that absorption takes place in the small intestine. So by the time the food reaches the large intestine, all the useful nutrients have been absorbed, and what is left is only these four. That is the cellulose, you can call them fibers or roughages, the greens that we eat in our diet. These ones are not broken down because we don't produce cellulase enzyme that can break down these fibers. And also, not all the food that you eat can be digested. Some will remain undigested and form what we call indigestible remains. And then some small amount of water will remain, and dead and living bacteria. These living bacteria are always found in the large intestine. Then these ones are the microbes which were killed by hydrochloric acid in the stomach. So these ones come about as a result of eating food that is already contaminated with microbes. Then in the first section of the large intestine, which we call colon, most of the remaining water, this water here, is absorbed. And once the water gets absorbed, this food starts to change from being a solution to semi-solid. All the water is not completely absorbed, but most of it. That's why the food is changing from solution to semi-solid material. And that material is what we call feces. Then these feces are going to be temporarily stored in the rectum until they get expelled out through the anus in the process of egestion. So we have this structure of the large intestine, as you can see here. The small intestine joins to the large intestine here at this section here. Then we have this ascending column, transverse column, and the descending column. So food moves through this direction, as you can see, and as it is moving, water is being absorbed. So by the time it reaches here in the rectum, it has become semi-solid. So it is going to be temporarily stored at the rectum here, and then later it will be expelled out in the process of digestion.